So today I will talk about convex neural codes in networks and sensory systems. So this is a very rough plan of my talk. First, I will tell you what I mean by convex sensing and convex codes. Then I will talk about recurrent networks and how convex codes somehow naturally arise in recurrent networks. And then towards the end, I will also tell you how convex codes and convex sensing can be exploited to infer something about neural systems that we don't understand well. Okay, so let me start with a statement most of you are well familiar. So neural activity in many sensory systems reside on low-dimensional manifolds, and the receptive fields are often convex on this manifold. So let me, instead of explaining what these words mean, I will just give you examples. So, and before I go on, let me just define one object, that is the receptive field itself. So let's say we have a single neuron, and X is a space of relevant stimuli. So then a receptive field is simply a function that takes a point in the space and produces, let's say, a non-negative number, which is, say, firing rate. Okay, so you can think of this as a function of stimulus space into the values of firing rates, right? And of course, if you look at well-understood system, you know what your relevant stimuli are. You can measure firing rates from experiments, so you can easily compute receptive fields. We are correlating the known stimuli and neural activity. So this is, of course, a classical example back from the 60s, V1 orientation tuning. And as most of you are well familiar with, there is a cat watching a movie, and it is shown bars at various orientations. And now here the stimulus space is literally a circle, just the orientation of a bar. And then, of course, measuring the firing rates of the single neuron, Hubel and Wiesel inferred this nice curve. And as you see, it's nicely unimodal. And on slightly more mathematical language, this is a concave function. And so here again, X, the stimulus space is just a circle. The function is, oops, sorry, I apologize. I need to remove this. Yeah, and the function is concave. Okay, so okay, another well-known example is place fields in hippocampus. So this is a structure in mammalian brain that is responsible for spatial navigation, episodic memory, and quite a few other things. And let me remind you the basic setup. So if you let a rat wander in two-dimensional environment, and measure firing rate of individual cells as a function of position. So you see this square over here. This is, say, just a rectangular room. And now what you see is the firing rate of individual neurons. And you see this, this is the heat map of the appropriate receptive field. They're called place fields. And as you see, again, they're nicely unimodal, or in more mathematical language, this is called convex. So coincidentally, this is another work that received Nobel Prize. So, and yet another example of receptive fields. This is internal cortex. And again, the well-known result that if you have the same setup, if you let the rat wander two-dimensional environment, and again, you record position of the animal and the firing rates, you get this this map, right? So, and at first it looks like, okay, this is clearly not unimodal and therefore not convex. So it sounds like I'm showing you a counterexample, but actually a very nice observation that was, I believe, also made in this paper from 2005 is that this is a regular hexagonal grid. So now if you wrap it into a torus, all of a sudden you obtain again a convex receptive field, just not on a two-dimensional space, but rather on a torus. But still, the basic pattern of convex unimodal receptive fields is nicely preserved even in so-called grid fields, which at the first look don't look convex at all. Okay. So, and coincidentally, all of the three examples I showed you received Nobel Prizes. So this is sort of the most publicized neural systems, but there are lots of other neural systems where the same pattern of convexity of receptive fields is preserved. So, and again, hopefully I convince you that neural activity is represented via convex receptive fields in many sensory systems. And on more mathematical language, they're called by different names: concave, convex, quasi-concave. This is not important. Okay, so, and there are some natural questions. If convex receptive fields are so ubiquitous in sensory systems don't live in a vacuum, right? So they are the output of most sensory system is a result of activity of some recurrent neural network in cortex, hippocampus, etc., etc. And somehow, if we look at sort of slightly averaged activity, they seem to be a result of convex receptive fields. Here is a natural question: how do neural circuits enforce convexity of neural codes? And that's question number one, which I will attempt to answer. And question number two, if assume we don't know anything about the stimulus space and receptive fields. So let's say we are not in hippocampus where quite a few things are known, but in some lesser studied neural system, let's say olfactory bulb. And assume we can still measure neural responses. Can we tell if, first of all, these responses come from convex receptive fields? And second, if can we say something more detailed about the stimulus space, like the dimension of the underlying stimulus space, for example? Okay, so of course, it's nice to ask questions, but it's entirely different matter of how we can answer them. So for the first, I will need the following simple tool. We call it the combinatorial code. Let me explain. So let's say we have a neural population. What is depicted here is just a raster of neurons, right? And this is very standard procedure. If you introduce certain window, you can get literally you can have zeros and ones. Basically, neuron was silent and neuron was not silent, right? So for each of these bins, you get a string of a binary string, which represents just activity of neurons. So some neurons were on, some neurons were off, right? And mathematically, you can think of this combinatorial code as just a subset of the power set. And they are known under different names. In computer science, they're usually called hypergraphs. It's basically a collection of subsets of the set 1 through n, and here was the number of neurons. Okay. So far, I have not said much. I just defined what I mean by combinatorial code. Now, if we look at the pattern of receptive fields, let's
Vladimir,
say, yeah.
Vladimir, exceptionally, I am interrupting you because there is a question of clarification that comes from Vincent Hakim. He would like you to make more precise the definitions that you give to concave and convex. So
okay,
yeah.
So convex means that the a subset of Euclidean space is convex if you draw a line between any 2 points in that space, and that line sits inside the set. So think of circles, ellipses, so whatever n-gons so that you draw a line between 2 points, and this line completely sits inside that subset, right? So here on this picture, actually, you see this. Let's say here, this is red spot is exactly what this subset. So if you draw a point in between two lines, sorry, that way around. If you draw a line in between 2 points, the line would sit inside this red region, right? So hopefully I answered that question. And concave function is a function whose sublevel sets are convex. So it's a bit technical, but basically this picture explains it all.
So let me just again, sorry. So then Vincent says that I understand, but the tuning curve was concave.
Concave, yeah. So concave is sort of the inverse of convex. So this is a concave function. So as a function, you see this is a function on 2D, right? So so you should imagine third dimension, right? So this is this kind of bell-like structure. So this is a concave function, but this subset, the red subset, is convex as a subset of two-dimensional space. Did I answer your question?
So please continue.
Yeah. Okay. Okay. So now, if we have receptive fields, then what happens is that let me just plot many receptive. This is just obviously a cartoon. So if you plot many receptive fields, we see that they split the stimulus space into regions that keep track of which receptive fields were covered in that particular subregion. So, and naturally generates a combinatorial code, right? So it generates exactly those subsets of neurons that are allowed to co-fire, right? And so translation, every set of receptive fields automatically generates the combinatorial code. And now, the first observation is that actually not every combinatorial code allows a presentation by convex receptive fields. So this is perhaps the simplest possible example of this happening. So here, the code is literally just one neuron fires, neuron 1 fires alone, neuron 2 fires alone, and neuron 1 and 3 fire together, neuron 2, 3 fire together. So this is just a nice depiction of how one would arrange the receptive fields. And here you see the receptive field number 3 is forced to split into 2 separate pieces. So this is clearly not convex. It's not unimodal, right? And one can prove that generically this code cannot be represented by convex receptive fields. So this is just a quick example to show that, okay, not all codes are born equal. Some codes can be represented by convex receptive fields and others cannot. And then, okay, there is quite a lot of mathematical formalism that was developed around this simple idea. So, for example, this little mathematical curiosity can be generalized with a lot more machinery to define so-called local abstractions. One can tell if codes are not convex, but I'm not going to talk about it. More importantly, what I would like to know, can we tell if the code is necessarily convex? And there is a theorem we proved a while ago that basically says that any maximal intersection complete code is convex. So maximal intersection complete code is something relatively simple. It means that if we take maximal partons, here maximal part, there are 2 maximal partons in this example. For example, it's 1, 3, and 2, 3, and take the intersection. The code is in max intersection complete if the intersection always lies in the code, and it's not intersection complete when it's not. So here you have, oops, I apologize. So here you see 2 examples. There is one code and there is another, and they differ by just on this one code word. So here it lies in the code and here it does not, and here it can be clearly represented by convex receptive fields, whereas here it cannot. All right. So now, so far, I just want to take this, remember this slide. And now, of course, here is a natural question. Okay, now let's connect it to recurrent networks. And a somewhat surprising result is that a generic recurrent network that satisfies the Dale's law indeed outputs convex code. So now I will try to explain this claim. So we consider a recurrent network that satisfies the Dale's law. Of course, this means that there are excitatory and inhibitory neurons. So here excitatory are in blue, inhibitory are in red. And the network receives only excitatory inputs. So the assumption is that only the long-term projections are all excitatory, as is common in cortex, right? And the only the excitatory output can be read out from the network. So again, clearly these assumptions are modeled after cortical columns, because this is exactly what happens on the level of cortical columns, right? And we look at a very concrete description of these networks. We just consider firing rate models with threshold linear nonlinearity. So here, this bracket plus is the standard ReLU function, right? So this is fairly standard firing rate model of recurrent networks. And here, how do we obtain a combinatorial code from this? Let me explain. So first of all, let's say we have a vector of firing rates in this network. And what we can do, we can record excitatory support. What is excitatory support? We literally look at the vector of neurons and look only excitatory neurons, not inhibitory. So look at each excitatory neuron and check if it is active or not. That is, if the firing rate is positive or zero, and record the subset of neurons that is active at any particular time. And of course, this is too much to us because there is a continuous dynamic. So what we will do is cheat. Let me explain how we cheat. So what we actually do is we look at the excitatory supports only at the fixed points or steady state. So the assumption is that the network obviously evolves. So it receives certain particular input from outside of the network, then it evolves. Let's say it goes to some steady states. And all we do is carefully recording the supports at this of excitatory neurons at the steady states. And this is exactly support plus, right? And then we define a code of such a recurrent network as a collection of all possible supports under all possible inputs. So here, this funny symbol denotes the fact that we consider all possible Bs, so all inputs to the network are valid. And all we do, we record excitatory supports at the fixed points, or there is a variant of this definition, so-called stable combinatorial code, where we record support at steady states as opposed to just the fixed points. Remember, in these networks, not every fixed point is automatically a steady state. There is stability analysis that needs to be done. So we distinguish these two versions of the code. All right. Okay, so so far this is just the definition of combinatorial code of a network. And again, it's to every synaptic matrix W, we can associate combinatorial code and the stable combinatorial code. And the goal, of course, is to understand the structure of the things in terms of W. And it turns out, oops, sorry, what happened? Okay, it turns out that the structure of this code can be completely described in terms of connectivity of the network and some other ingredient, which is spectral in nature. So first, let me describe the relevant connectivity features of these networks. So let's say this is the network we consider. This is just a cartoon depicting whatever connectivity that happened here, right? And we collect two objects. One is excitatory connectivity graph. We denote this as GE, E for excitatory, which basically takes all the blue connectivity and strips out all the red. Red here is inhibitory, blue is excitatory. So it's very simple. So it's excitatory subnetwork connectivity. And there is another feature of this connectivity that we need. We call it beta. This is the set of excitatory neurons that do not receive recurrent inhibition. So presumably not every excitatory neuron in the network has to receive recurrent inhibition. So in this picture, these are neurons 4 and 5, right? So they don't receive recurrent inhibition. So there is this very special set of excitatory neurons that doesn't receive inhibition. Let's call it beta. Okay, now we take these two features and assemble them into this object, which we call code of G and beta. So again, this object is associated with connectivity and nothing else of this network. It doesn't take into account particular values of synaptic strands. And what this says is that this is sigma belongs to the code if all the synaptic targets, according to the excitatory subnetwork, the uninhibited pieces of this lie back in sigma. So this is a very special combinatorial condition. And it turns out that this is the only feature of connectivity of this network that matters for fixed points. So if you care for dynamics, the story is much richer. But if you care just for the fixed points, this is all that one needs to know. Okay, so the first theorem is a complete characterization of the code of the recurrent network. Basically, it says that the code of the recurrent network can be described as follows. It's sigma, which is a subset of neurons or excitatory neurons, belongs to the code if there are two conditions that satisfied. One is sigma is in this object that was defined on the previous slide. So it's completely determined by connectivity alone. And another condition is a spectral condition on some excitatory subnetwork where beta sigma are the neurons that of sigma that don't receive recurrent inhibition. So this is mathematical description of this. And here W beta sigma are synaptic weights of the network among uninhibited neurons in sigma. And rho is just the spectral radius of this subnetwork. So this is complete characterization of the code, which is really nice. And here it still cares for values of synaptic efficacies, but only on this very peculiar sort of detail of this. And otherwise, it's just all about connectivity. And moreover, there is a so-called weak Kaplan regime. That is, if we assume that the synapses are sufficiently weak, that is, the Frobenius norm of the synaptic matrix is smaller than 1, then it turns out that in this weak Kaplan regime, the combinatorial code is completely determined by the connectivity features. And moreover, the combinatorial code is the same as stable combinatorial code. That is, every fixed point is a steady state. So the fact that every fixed point in this regime is steady state, I presume it was well known before. There is quite a literature on the subject, but to me, this feature that stands out that now it can be completely described. This is just connectivity features of the network, right? So it can be completely described in terms of connectivity features of the network. So this is a really interesting theorem. And a corollary of this is actually one can now play with this combinatorial description relying on the connectivity features and answer this question in earnest. Remember, what I told you is that some combinatorial codes are not convex. And there was a question, okay, but wait a second, how do so many neural systems output convex codes nonetheless? And here is the answer. So a generic recurrent network that satisfies Dell's law actually outputs convex codes. And the way we proved it was proving that property of the code being max intersection complete. So it relied on our earlier work about convex codes. So, and this is very brief version of this theoretical development. So if you are curious, you can find this in this preprint we put on archive. All right. So now I basically finished telling you this part of the story. Again, I hopefully somewhat convince you that codes of recurrent Dale networks are convex, that is, are compatible with convex receptive fields. And now I'm going to this more illustrative part of my talk, where I will try to illustrate how convex sensing can be exploited in new sensory systems that are not fully understood. And my favorite example here is the sense of smell, olfaction. The problem with, or rather the difficulty with olfaction is that unlike many other sensory systems, the stimulus space in olfaction is extremely poorly understood. Of course, it's this huge mass of chemicals, and presumably it's extremely high dimensional. But what people do know is that whatever animal sense of olfaction throws away most of this information and projects it into presumably much smaller dimensional space. And then the next question, okay, if we somehow assume that the receptive fields in that new space are convex, can we infer something about the space? For example, the dimension of olfactory space, which is still somewhat controversial topic, as a matter of fact. Okay, and question: can we exploit convexity of receptive fields to learn new things about neural representations? So, and here we actually use some data that comes from the lab of Matt Vakovic at University of Utah. And again, what they do is calcium imaging of olfactory bulb. So what you see is actually surface of olfactory bulb in response to particular odorants. And so for people who don't know much about olfactory bulb is split into many glomeruli, which express, each glomerulus expresses a unique olfactory receptor that has its own response to particular chemical spectrum. And you see this glomeruli as these blobs here. In the mouse, there are over 1,000 such glomerular types, rather. And what people can measure is this response map. So basically, you present whatever odorants, and you receive olfactory responses. And again, we can measure stimulus glomerular response. Of course, you can measure the chemical descriptions, which we actually won't use. So what do we want to know? We want to know what are the natural coordinates on the olfactory space. So regardless of what the chemical composition, one can hope to find sort of ideal description of the olfactory space. And what is the geometry of the olfactory response, if possible at all? And the question we don't intend to answer at all is how do physical-chemical features translate into this natural coordinates? This is not what I'm going to talk about. Okay, so sorry, 1 second. Yep. Okay, so now we have the chemical space. So each space is an odorant or a combination of odorants plus concentration. And there are glomerular maps, so that translate this into neural activity space. This is how each glomerular type relates to other glomeruli. So how do they fire? And what we want is to infer the dimension of this neural activity space and maybe explore some basic properties of this space. So how do we do this? So the idea is roughly the following. So we assume that the receptive fields, whatever the space is, if you just postulate that receptive fields in this space are convex, that actually imposes constraints on the dimension. So that's the idea. And then, of course, we need some model here, which I will describe it briefly here. And my model will be extremely rudimentary. So let's say we have a d-dimensional olfactory space. And let's say there are natural coordinates there. Let's say just Euclidean space. There are d natural coordinates. And let's say each particular olfactory receptor does something extremely simple. It's literally a linear function passed through some unknown nonlinearity. So here, G, this nonlinearity G is unknown, but what we do know, it's monotone increasing function, right? And so this is just a picture that illustrates what can happen. So if this is our olfactory space, So the points here are particular orderons, and let's say G is some nonlinearity. Here black is 0, and here something that happens to be non-zero. And if this is W, vector W, then you will see this kind of response. And as you see here, each receptive field is trivially convex because it's basically half space. So here it automatically falls under this in this characterization of convex receptive fields. So of course, none of this is observable. So the only thing we can observe is the response of each glomerulus. And if it was one glomerulus, of course, I would be lying if I told you that anything can be learned from this. But luckily, we don't have just one glomerulus. We have many. So question, can we infer any useful information about the space of glomerular response from the observables, which is just response itself. So this is this matrix of glomerular versus orderons. And the answer is yes. So it relies on some machinery of algebraic topology, which I'm not gonna do any justice due to time constraints, even though I can point you to a manuscript where it is described. But let me quickly explain the idea. So one starts with this matrix and produces so-called Betti curves, which basically which detect this is like some topological summary of a profile of the space in each dimension. So here, beta 1, beta 2, beta 3, 1, 2, 3 correspond to different dimensions of space. And if the space, say, is three-dimensional, that this Betti curve should be non-zero. And the machinery is a bit more complicated. So here you see there is so-called density, which I'm not going to explain in any detail, but basically you threshold this matrix at certain thresholds and construct a sequence of simplicial complexes, which is an object from algebraic topology. And from the simplicial complexes, you compute so-called Betti numbers. Again, this is a topological invariant. And the cool part about this topological invariance, so if you remember the original formula for the model, there is this unknown nonlinearity. And of course, if not for this nonlinearity, this problem would be entirely trivial. You literally do SVD and you're done, right? So the trouble is this nonlinearity, which can completely destroy whatever linear rank you might want to compute. And here, what this machinery does, it is invariant with respect to applying any monotone function. So if you apply, if you had this vectors W's and X's and applied one nonlinearity and applied another nonlinearity, you would get exactly the same Betti curves as the output. So there is no difference. So the details of this are explained in this manuscript, which so I didn't do any justice. So now, so how does this look like on real data? So one can compute Betti curves, let's say for the real data in the olfactory bulb. And you could see that, here, how do we know that the dimension is, how do you know that the dimension is larger than 3? So here the real data is these punctured lines, and this is a simulated data in dimension 3. So in dimension 3, there is not much. Sorry, in dimension 2 rather. Sorry, in dimension 2 there is clearly no three-dimensional features. So here these curves become 0, but for the real data they're very non-zero. So we know that the data is certainly not two-dimensional, which is of course not a big surprise. So, but this is in dimension 5. So one can do similar simulation in dimension 5, and now you see that this Betti curves roughly match. So, and it turns out that okay, this analysis is just an illustration. So this can be a lot more statistically rigorous. I skip all the details, and there are actually two models one can consider. One is no inhibition, which is that all the dimensions are born, all the directions are born equal. And another is, inhibition is allowed, that is all the directions are born equal in the olfactory space, and non-inhibition, that is some directions are preferred than the others. And one can look at these Betti curves and design a summary of these Betti curves just as a point in two-dimensional space. Let's say this is beta 1, beta 2. We literally take integral under this curve, and this obtains as a point. And this is the summary that one obtains from this. So this is the colored boxes and dots are artificially simulated data in various dimensions. And the black dot is the actual data. And as you can see, first of all, this analysis immediately separates two different hypotheses. One is no inhibition, that is, not all directions in the olfactory space are born equal. So this is very anisotropic. And this is allow inhibition, which means that the space is anisotropic. So the data clearly lies in this region. The real data really lies in this region. So not all directions in olfactory space are born equal. And moreover, one can roughly estimate the dimension. So here dimension is consistent between d equals 5 and d equals 7. Unfortunately, we cannot be more precise because there is not enough statistical power in this analysis. Okay, and I'm almost close to the end of my time. So what about the geometry? Can we infer the geometry of olfactory space? So we also design an algorithm that actually unravels this formula with unknown nonlinearity and still infers the position of the of particular odorants in the olfactory space. So what you see here is the following analysis. We took this matrix of the stimulus response matrix. So then we used our method to unravel this formula to obtain the positions of this XA, and then we separated them by chemical class. So here you see yellow is acids, orange is aldehydes. Actually, I'm terrible in chemistry, so there are kinds of chemical classes. And as you can see, these chemical classes nicely cluster in this very abstract olfactory space. So which means that this olfactory space has something to do with reality, because the original responses were also kind of clustered around the particular chemical classes. So this shouldn't be a surprise. So I think I ran out of time. So this is the end of my talk. So people who were most involved with what I talked about, this is Nicola. He did all the hard work regarding the analysis of recurrent networks, and the olfactory story. There were two people involved, Minchun Wu and Yasha Berchenko-Kogan, and also the data was obtained from the lab of Matvakov.